In 1994, the search for the immortalizing gene known as telomerase was well underway at a biotech company called Geron Corporation in Menlo Park, California. If the technology worked, it could reverse the aging or even immortalize human cells. That should be an easy matter with cells cultured in the laboratory dish. But how could we use it to treat the actual aging of human beings? I took a few minutes to walk along the San Francisco Bay to shake loose my thoughts on the matter. Turning the telomerase gene on in somatic cells of the body would indeed be a type of immortality transfer, but it would be technically difficult. It would likely require a form of gene therapy, a challenge to implement in those days. Perhaps I thought we could immortalize a type of stem cell, like the blood-forming stem cells in the lab, and then reintroduce them into the body. That could conceivably allow us to reset the lifespan of the immune system in aged patients. But could we do something even more profound? My thoughts turned to a type of cell that had always entranced me from the first time I'd learned about them. The mother of all stem cells, extracted directly from the germ-line. While only isolated from certain kinds of mice, these cells seemed almost magical to me. In the 1960s, a scientist named Leroy Stevens published the first studies about these cells, isolated from the 129 strain of mice, capable of forming all the tissues of the body. His scientific curiosity was triggered by the apparent capacity of the cells for immortal proliferation, as well as their power to become any of the cell types of the body. As a result, he referred to these cells as pluripotent, from the Latin word plurimus meaning many, and potens meaning power, embryonic stem cells. The cells were cultured from the immortal germ-line. The embryonic stem cells, shown here as ES cells, normally had several choices. They could continue along the immortal cascade into embryonic germ cells, or shown here as EG cells, and then after that the sperm or egg cells, continuing the immortal propagation of the species. Or they could steer into the right and become mortal cells of the body. Unlike other germline cells, though, like sperm and egg cells, mouse ES and EG cells had the potential to proliferate endlessly, and they showed that property in the laboratory dish. They came from the egg cell just a few days after it was fertilized, in particular from the cluster of cells called the inner cell mass. These cells were in the process of dividing, and since they still had the power to steer left or right, that is, to continue the germline or to make a body, they had these twin powerful characteristics of pluripotency and immortality. Any cells derived from them would be born young, in the same way babies are always born young. When cultured in the laboratory dish as an expandable cell line, they opened the door to do some really amazing things. Soon after Stevens's publications, other scientists began to build upon his pioneering work. For example, the twin powers of pluripotency and immortality opened the door to what is called mouse transgenesis. Some of the key players in this were later to receive Nobel Prizes. They included Martin Evans, Oliver Smithies, and Mario Capecchi. These scientists and others built a technology platform that allowed any precise change in the blueprint of life, known as DNA, to be engineered into mice. And here's how it works. Mice are allowed to breed, and the fertilized egg cell of a female is allowed to develop, but only for a few days, so that it's still a microscopic cluster of unformed cells. Normally, these fertilized egg cells travel toward the uterus. If they attach there, they would then become embedded into the uterine wall and then begin the process of what is known as differentiation. In other words, these unformed cells begin to branch out into what will eventually become all the cell types of the body, the somatic cells. If the fertilized egg cell does not attach, no pregnancy is initiated and no body cells are formed. Following Stevens's lead, the scientists cultured the inner cell mass cells. It was seen that these cells could be expanded in number and maintained in this blank and unformed state, such that they did not differentiate. The cultures were then called mouse pluripotent embryonic stem cells, or ES cells, after the tradition of Stevens. Now, since the mouse ES cells can be cultured in the laboratory, it was a relatively simple matter to modify their DNA. Basically, a piece of DNA is designed to have a particular effect. For instance, to target or remove a gene from the cells, or perhaps to add a gene even from another species, such as the fluorescence gene from jellyfish that make them glow in the dark. Then the genetically modified ES cells are injected back into the blastocyst and implanted into the uterus of a mother. The result, typically after some breeding, are mice that are now displaying the effect of that genetic modification. If the jellyfish fluorescence gene is used, the mice will glow in the dark. Because this allows the exchange of genes across species, the technology came to be known as transgenesis, and the mice as transgenic mice. Now, what if such powerful cells from the human species could be captured in the lab dish and expanded in numbers? Such cell lines would represent the immortal human germ-line, the very cells that provide the power of producing coming millions of humans. Instead of using them to make transgenic humans, which certainly would have ethical issues, they could instead be used to solve the largest unsolved problem in medicine, namely, a means to replace worn-out cells and tissues in the body with young counterparts, a vast, cost-effective parts supply store for the human body. I suppose the analogy would be that of keeping old automobiles on the road. If you could imagine a world where we used automobiles but never had invented a means of repairing them, then it would be basically impossible to use a car indefinitely. After 100,000 miles or so, the tires would wear out or the motor would fail. But a cost-effective means of manufacturing the components of the car would allow the car to be remodeled. Despite the fact that the lives of our loved ones are immeasurably more valuable than an automobile, we have never found a means of manufacturing to scale all the replacement parts of a human being to keep us on the road, so to speak. Today, when someone has a heart attack, it can destroy heart muscle, and there is no therapy to restore those cells. That's why heart disease is the #1 killer today. In the case of Parkinson's disease, cells are lost in the brain. If they could be restored, it would cure the disease. The same is true for diabetes, or age-related macular degeneration that robs so many people of vision, osteoarthritis that causes so many people pain as a result of age, and so on. If we could make transgenic mice with ES cells, we could potentially combine telomerase and human ES cell technology to make immortal human cells of any kind. But could human embryonic stem cells be isolated and cultured in the lab dish? I realized that such a project would have issues. That was clear from the start. I knew the field of cell therapy as a modality to treat aging had a checkered history. In the previous video, I described the Erector Set, a group of clinicians that attempted to use the power of the germline to rejuvenate patients. After Serge Voronoff's patients reportedly contracted monkey venereal disease, a protege of Voronoff named Paul Nihans used fetal sheep tissues, cells that should not be contaminated with bacteria or viruses. He minced up the tissues so that the cells could be easily injected with a syringe, and he called it cell therapy. The problem with using animal cells is that they would almost certainly be immediately rejected by the patient's immune system, but that didn't slow down Nihans. Since he could extract any tissue from the fetal lamb, he had a story to tell that would arguably treat virtually any disease. If you had heart disease, he would mush up the fetal heart cells and inject them. He had uncovered a gold mine, or perhaps we should say a mine for fool's gold. And combined with the promise of reversing aging, he became a founding member of the rich and the famous. His fame was elevated even more with the opportunity to treat Pope Pius XII. The Pope became critically ill in 1954, and his staff reached out to Dr. Nihans, hoping for a miracle, and they found it in the science of the lambs. The Pope did recover shortly after getting a series of shots, and in gratitude he granted Nihans membership in the Papal Academy of Science, a title he proudly displayed on his letterhead. He was flooded with patients. It was estimated that he treated tens of thousands, and a whole industry grew in competition. But careful scientists did not buy the undocumented reports of success. The cells died after being injected, that much was certain. Animal cells would rapidly be identified as foreign and rejected by the immune system. So these previous incarnations of cell therapy had somewhat tarnished the field. And then there was another potential issue. In the late 1970s, there had been a considerable uproar over what is today called in vitro fertilization, or IVF. First developed by Patrick Steptoe and Bob Edwards, IVF began with intense controversy. While cherished by the parents, the children were branded by the critics as test tube babies. In the course of a typical IVF procedure, numerous pre-implantation embryos are made, and the best ones are implanted in the mother. The others are often frozen. If the mother becomes pregnant, the excess ones are commonly incinerated. Rather than destroying these blastocysts, wouldn't it be preferable to retrieve them before they were destroyed and use them to make permanent ES cell lines? Only a handful of these lines would likely be necessary to enable the entire field of regenerative medicine because the cells would replicate indefinitely. This all seemed very logical to me, but I knew that people brought a lot of emotion to the issue of abortion, and this technology, while not the termination of a pregnancy, would likely get caught up in the debate, especially by those with little or no interest in learning the facts. Despite these concerns, we at Geron organized a collaboration with 3 scientists with access to discarded embryos in the hope that for the first time in history, we could isolate these potentially immortal and pluripotent cells. The first collaborator was Dr. Roger Peterson, then a professor at the University of California at San Francisco and director of their IVF program. The second was John Gearhart at Johns Hopkins School of Medicine, and the third, James Thompson, then at the primate facility at the University of Wisconsin at Madison. In 1995, Jamie helped us start our work with ES cells that he had isolated from non-human primates. But the big breakthrough came in early 1998 when all 3 labs began to show success in the isolation of human ES and EG cells. As Roger was to say in one private meeting, "Gentlemen, something magical is about to happen." I had the same sense. The ability to manufacture any human cell type genetically modified in any way, while simultaneously granting the power of immortality to these cells, opened the door to an immense field of opportunity, only limited by the human imagination. On hearing of the cells, William Haseltine, formerly the CEO of Human Genome Sciences, called the emerging field regenerative medicine, and that name quickly took hold. The public first became aware of these cells on Friday, November 6, 1998, after papers on both the ES and EG cells were published in the scientific literature. As Nicholas Wade wrote in his opening paragraph, pushing the frontiers of biology closer to the central mystery of life, scientists have, for the first time, picked out and cultivated the primordial human cells from which an entire individual is created. As one might predict, some members of the pro-life community mistakenly equated ES cell technology with abortion. Harold Varmus, a Nobel laureate and head of the National Institutes of Health, supported federal funding for the science, saying, "The development of cell lines that may produce almost every tissue of the human body is an unprecedented scientific breakthrough. It is not too unrealistic to say that this research has the potential to revolutionize the practice of medicine and improve the quality and length of life. Nevertheless, 70 members of Congress proposed legislation that would ban any such use of federal funds. Shortly after that, 73 scientists, of which 67 were Nobel laureates, countered in a letter to Congress urging that Varmus's position on the cells be respected. When President Bush came into office, he halted all federal funding for the cells, and then, on August 9, 2001, announced his position on the matter.
Good evening. I appreciate you giving me a few minutes of your time tonight so I can discuss with you a complex and difficult issue, an issue that is one of the most profound of our time. The issue of research involving stem cells derived from human embryos is increasingly the subject of a national debate and dinner table discussions. The issue is confronted every day in laboratories as scientists ponder the ethical ramifications of their work. It is agonized over by parents and many couples as they try to have children or to save children already born. The issue is debated within the church, with people of different faiths, even many of the same faith, coming to different conclusions. Many people are finding that the more they know about stem cell research, the less certain they are about the right ethical and moral conclusions. My administration must decide whether to allow federal funds, your tax dollars, to be used for scientific research on stem cells derived from human embryos. A large number of these embryos already exist. They are the product of a process called in vitro fertilization, which helps so many couples conceive children. When doctors match sperm and egg to create life outside the womb, they usually produce more embryos than are implanted in the mother. Once a couple successfully has children, or if they are unsuccessful, the additional embryos remain frozen in laboratories. Some will not survive during long storage. Others are destroyed. A number have been donated to science and used to create privately funded stem cell lines, and a few have been implanted in an adoptive mother and born and are today healthy children. Based on preliminary work that has been privately funded, scientists believe further research using stem cells offers great promise that could help improve the lives of those who suffer from many terrible diseases, from juvenile diabetes to Alzheimer's, from Parkinson's to spinal cord injuries. As the discoveries of modern science create tremendous hope, they also lay vast ethical minefields. As the genius of science extends the horizons of what we can do, we increasingly confront complex questions about what we should do. Research offers hope that millions of our loved ones may be cured of a disease and rid of their suffering. I have friends whose children suffer from juvenile diabetes. Nancy Reagan has written me about President Reagan's struggle with Alzheimer's. My own family has confronted the tragedy of childhood leukemia. And like all Americans, I have great hope for cures. As a result of private research, more than 60 genetically diverse stem cell lines already exist. They were created from embryos that have already been destroyed, and they have the ability to regenerate themselves indefinitely, creating ongoing opportunities for research. I have concluded that we should allow federal funds to be used for research on these existing stem cell lines, where the life and death decision has already been made.
The growing debate about the use of ES cells was fueled in part by a bombshell announcement on February 23, 1997. A group of animal science researchers at the Roslin Institute in Edinburgh, Scotland, led by a scientist named Keith Campbell, reported for the first time that a mammal, in this case a lamb, had been cloned from a somatic cell. This astounded scientists because until that time, everyone believed that the changes that cells undergo in leaving the germ-line to become somatic cells was irreversible. Campbell's team had taken an epithelial cell from breast tissue and transferred it back into the germ-line, in this case an egg cell, and then transplanted the reconstructed embryo back into the uterus. What was once a cell from breast tissue was now transported back in time to become the beginnings of life, to be born a second time. Because all of the DNA, the blueprint of life, came from the original animal, the lamb was identical, or a clone, of the original. The cloned lamb was named Dolly, after Dolly Parton, since she owed her origins to breast tissue. Of course, the world latched onto the amazing science of cloning. Stories immediately began to circulate about the possibility of cloning human beings, and there was speculation that scientists would soon clone Hitler, the Beatles, or some billionaire. Perhaps the most bizarre story was a live press conference from Hollywood, Florida, where Brigitte Bosselier of a religious group called the Raelians stated, "I am very pleased to announce that the first baby clone is born. She is fine." But where was the child? On the theory that the reported clone could have associated health risks, Bernard Siegel, an attorney with a history of advocating for children's rights, filed a petition in juvenile court claiming that the supposed child needed a court-appointed legal guardian to assure his or her well-being. Needless to say, the Raelians never produced the cloned child. But some of us were interested in quite a different aspect of this miracle of cloning. Would the transfer of an aged somatic cell into an egg cell not only transport it back in developmental time, but also reverse aging itself? Or would the clone be born as old as the original animal? To put it another way, would the clone really be a lamb, or would it be a sheep in lamb's clothing? I was of the opinion that the transfer of an aged somatic cell into the germ-line, in this case the egg cell, would actually reverse aging. I, for one, have always objected to cloning humans, but if it were done, I believed that if the cell came from a 100-year-old person, the baby would still be born young and would have a normal lifespan. And if that clone was cloned, its clone again would have a normal lifespan. In other words, the process of cloning, first the removal of the genetic information from the egg cell using a fine glass needle under a microscope, and then the transfer of a somatic cell into the egg cell, was in fact performing a near miracle of cell biology. It was a cellular time machine, fully capable of reversing the aging process. Then, if the resulting blastocyst was used to make an ESL line instead of making a pregnancy, it could be an endless source of young replacement cells, all of which would be identical to the patient. This came to be called therapeutic cloning, to distinguish it from the cloning of babies. So the burning question in those years was whether SCNT would not only restore pluripotency to a somatic cell, that is, reverse development, but reactivate telomerase and reset telomere length as well. I chose to be associated with a U.S. cloning company called Advanced Cell Technology, or ACT, while my old company, Geron, collaborated with the Roslin Institute. The Roslin Institute reported in 1999 that this idea wouldn't work. In the journal Nature, they reported that Dolly's telomeres were very short, suggesting that cloning did not reset the telomere clock of cell aging.
Is Dolly the sheep mutton dressed as lamb? That's the controversy surrounding today's findings. They show at almost 3 years old she looks young and fit, but the genetic material inside her cells appears to be much older. Even the scientists who created her don't know if that means she'll die young.
There is a possibility that as these changes continue in the cells, that she will have a shorter life.
At ACT, we were in the business of cloning cattle for agriculture and to make transgenic cattle to produce desired proteins in milk. We took cells from a cow, grew them to the Hayflick limit, and then used the senescent cells in a carefully controlled study of the ability of cloning to reverse aging. In the year 2000, we published our results that SCNT could not only reset telomeres back to the beginning of life, but we could actually transport the cells even further back in time to make animals younger than young. The cells that were once senescent now had a proliferative lifespan longer than the original cells. Recalling the ancient mystery religions, foreseeing the power of immortal renewal to cause life to be born again, we called the first such rejuvenated animal Persephone.
These cows may be the closest thing to time travelers the world has ever seen. Scientists at Advanced Cell Technologies in Worcester, Massachusetts, believe they've found a way in cloning these 6 cows to reverse the aging process. Their findings appear in the latest issue of Science magazine.
To our knowledge, this has never been seen before in science. We can take an old, tired cell and take it back in a little time machine, and that cell becomes young again.
That's quite the opposite of what happened to Dolly, the famous cloned sheep. Her cells showed signs of premature aging. But researchers here say they used cells from a different part of the body and a different cloning technique. They put old cow skin cells in an egg cell and watched them completely rejuvenate.
When other cows the same age are starting to grow old and frail, according to these data, those cells should, in theory, in these cloned animals, actually be still able to divide as many times as a newborn calf.
Researchers are not sure whether younger cells will translate into a longer lifespan for the cows. But even if the animals don't live any longer, just being able to figure out how to produce younger cells could have a profound impact on medicine.
This technology may allow someday us to make for a patient of any age a young cell of any kind that they might need transplanted to cure disease.
Researchers say this could solve the problems of heart disease, diabetes, osteoporosis, and the shortage of organ transplants. It could also conceivably create the fountain of youth.
An adult would have the immune system of a teenager. The reproductive years potentially could be extended to age 60, 70, or even 80.
The study's authors predict clinical trials on humans will begin in the next few years. They say the real question is not if science will be able to turn back the clock in humans, but when it does, will we be ready?
We decided to test it by transplanting young, blood-forming cells into an old animal.
Millions of Americans suffer from diseases like these, diseases that involve a faulty immune system. What if therapeutic cloning could fully restore not just organs, but a defective immune system? That's the goal of an ambitious experiment that begins in Pennsylvania. This cow is about 12 years old, no spring chicken. Researchers will try to replace her aging immune system with a peppy, rejuvenated system. Skin cells from the old cow are shipped to Advanced Cell Technology in Massachusetts. The old cow is cloned, and the resulting embryo is rushed back to Pennsylvania. The embryo is implanted in a surrogate mother, then removed and flown by charter plane to New York. The tissues are rushed to the Memorial Sloan Kettering Cancer Center. While the pilot stands by, immune system cells are extracted from the clone. Back in Pennsylvania, these cells are transfused into the old cow. A perfect genetic match from a clone, these cells shouldn't be rejected. The transfused cells travel through blood vessels. They should find their way into the old cow's bones. There, the immune cells should settle into the marrow, the core of the immune system. If it works, the marrow will soon start cranking out new white blood cells, the workhorses of the immune system. The Pennsylvania team takes regular blood samples from the old cow, which are shipped to the Mayo Clinic in Rochester, Minnesota. Here, the blood is analyzed. Are there fresh white blood cells? Peter Wettstein, an expert on the immune system, is about to see the results for the first time. He wants to know if the old cow's white blood cells are on the rise. The figures look good, but have the new white blood cells really come from the transfused cells? 10 days later, good news arrives at Advanced Cell Technology.
We
just got some new information that suggests that the new immune cells that we place into the old cows have definitely taken up home and appear to be producing white blood cells. And this data is preliminary, but it looks very encouraging.
For those who could see the implications of the science, it was clear that the next step was to apply the technology to the human species. Could we reverse the aging of human cells using SCNT? Could we cause an aged somatic cells to, in a sense, be born a second time, or Renatus in Latin? But where would all the human egg cells come from? At ACT, we had a steady and reliable source of cow egg cells. Could we use those? To track the human cells, we made them transgenic by adding the fluorescence gene of jellyfish, and then transferred those cells into cow egg cells. President Clinton asked his Bioethics Commission to weigh in on the matter. I suspect that he would even been more alarmed if he had known that the reconstructed embryos were, in fact, cloned, genetically engineered human embryos carrying the fluorescence gene of jellyfish, cloned in a cow egg cell. We heard that the joke floating around the Clinton administration was that Willie Nelson had already written a song about the technique, "Mamas, don't let your babies grow up to be cowboys." Comics compared the technique to the myth of Theseus and the Minotaur, with the thread guiding Theseus through the labyrinth being the DNA molecule. Finally, after years of preparation, we had put in place an ethics advisory board and procedures to begin our actual research on human SCNT using human egg cells. Despite what the press reported as bodyguards protecting us in the controversial research, the bodyguards were required by our Ethics Advisory Board to guard the egg cells to be sure that no one could misappropriate them for an unapproved use. During 2001, we had observed the first stages of success in the research. In reading the history of IVF by Steptoe and Edwards, we noted how they reported on each step of their research, first the successful fertilization of the egg cell, the first stages of cell division, and so on. So in 2001, we decided that we should publish in the scientific literature our progress to date. We even allowed the press to look over our shoulders, and we gave them the power of the pen to write as they saw fit, either favorable or unfavorable commentary. Our only request was that they not publish a story until after we publish the scientific results. After the process of peer review, our paper was finally ready to publish. I received a call about a week before by U.S. News & World Report that they were going to hold their normal publication day in order to break the news on the Sunday program Meet the Press. You should be there, they advised. Not knowing if we would be praised or vilified on the program, I agreed to attend. Fortunately, the story was focused on the promise of therapeutic cloning and was not highly critical. But not everyone agreed. Pope John Paul II was quick to condemn it, and President Bush criticized it as well.
Turning now to news at home. A biotech company in Massachusetts said today it has cloned a human embryo. The scientists say their ultimate goal is not to create human beings, but rather embryonic stem cells. That in itself is controversial. ABC's Dan Harris has this report.
It took a year and a half of painstaking, top-secret work at a small biotech company in Massachusetts called Advanced Cell Technology. None of the embryos involved in the research developed past the early stages, but the hope is that eventually cloned embryos can develop enough to provide large amounts of stem cells. Stem cells are the body's utility players. They can grow into any type of cell.
The concept is that we could do for the human body what it's so easy to do for an automobile, make replacement parts.
Put very simply, what the scientists did was inject DNA from an adult human cell into a human egg, which then grew into an embryo. It may still be years away, but scientists say human embryonic cloning could help patients with a variety of conditions, from damaged spines and hearts to diabetes and cancer.
While this may be a medical breakthrough, it is definitely an ethical dilemma. Some worry the inevitable result is that infertile couples will use human cloning as a way to make babies.
A bill banning all forms of human cloning has already passed the House. The author, Congressman and Dr. Dave Weldon, says the researchers who created Dolly first created sheep with severe defects.
If you try to create cloned human beings, you have the real possibility of babies being born with severe birth defects.
The scientists at Advanced Cell Technology, who say they do not support cloning entire human beings, argue the risk of abuse does not outweigh the reward, alleviating human suffering.
And to say, look, we're afraid of the future, and we're afraid of a brave new world, we're afraid to go help these people, would be irresponsible.
By publishing their methods, Advanced Cell Technology admits, despite its best intentions, it may have already provided scientists the world over with a human cloning cookbook.
It is important to note that at the time, the technique was the only known method to make young cells of any kind identical to a patient so that they would not be rejected. In my mind, it was the most potent technology to directly address the problem of human aging and age-related disease. But others, such as Leon Kass of President Bush's Ethics Commission, not only criticized human therapeutic cloning, but ES cell technologies, and in fact, any intervention in human aging. As reported in the New York Times, Dr. Kass has found himself setting special value on the natural human cycle of birth, procreation, and death. Just as he opposes most kinds of interference in reproduction, he is also against deliberate efforts to increase longevity. The proper goal of medicine, in his view, is to improve health, not to conquer death, which he sees as a necessary and desirable end. The finitude of human life is a blessing for every human individual, whether he knows it or not, Dr. Kass has written. This reminded me of the opening scene of a television show I used to watch as a child called Ben Casey. The show seemed to be portraying the role of the physician as a soldier, fighting the reality of the human life cycle, not to promote the cause of death, but to alleviate suffering and to fight for life.
Infinity.
But ironically, the pro-life party was setting itself up to advocate a pro-death stance. Death was good, they argued. We owe God a death. The controversy generated grassroots support and institutional opposition. On the grassroots side were patient advocates like Michael J. Fox, representing patients with Parkinson's disease, and Mary Tyler Moore, representing those suffering with diabetes. On the institutional side were pro-life congressmen like Sam Brownback or John Paul II, though some pro-life congressmen like Orrin Hatch studied the science and were ardent supporters.
The sands are running through that hourglass, and time is running out for people and their families that care for them. And we have an opportunity here to move forward with something. I mean, these are all good people. No one here is a mad scientist. No one here wants to do anything that is ethically or morally wrong. I think everybody's guided by the same principles and the same goal, which is to help people. We just think that those people are here now. They're waiting for answers. We have the ability to help them. We have the procedures to allow this funding to happen, and I just think there's no compelling reason not to. And if the argument is that argument about destroying the life of that embryo, that happens anyway on a large scale. Those cells are being destroyed on a daily basis, and by the president saying that certain cell lines can be available, he's already basically said, I think this is viable work. I think this is quality research and quality science. I'm just saying you can't do this much of it. You can do this much of it. Well, once you say we can do this much of it, what's the difference? You know, we're going to do it responsibly and ethically, and I just think that he's got an opportunity to really do something fantastic for the world, for the future of medical research and the health of this country and the people all over the globe. So I just ask him to, carpe diem.
It's been nearly 6 years since human embryonic stem cells were first successfully cultured by Dr. Jamie Thompson at the University of Wisconsin. Since this critical milestone, we have taken small steps toward unlocking the therapeutic potential of human stem cells. Yet during that same time period, diabetes has contributed to the deaths of as many as 3 million people. and cost our nation over $750 billion. It has caused nearly 500,000 amputations, rendered over 100,000 people blind, and forced 250,000 to require kidney transplants or dialysis. And 120,000 moms have been told that their child has type 1 diabetes, a disease which during that time period would require each of these children to have 8,700 injections of insulin and 17,500 pricks of their fingers to check blood sugar levels, just for that child to survive. And although all the steps forward are important in a field with so much promise and so many lives at stake, I believe it is our duty to craft policies and take actions that allow us to make progress, not by steps, but by leaps and bounds. Unfortunately, it has become clear that we will not achieve the dramatic advances that we all are hoping for unless we expand the current stem cell research policy. But like President Bush, I am pro-life. I also feel that we show special reverence for the potential life that resides in excess in vitro fertilization embryos by allowing them to be used to save lives in the future, rather than watch them be discarded as medical waste.
We're here because we believe that when science offers the door to hope, we shouldn't allow rigid ideology to slam it shut. A true culture of life seeks to cure disease and alleviate suffering, not deny help. That's why many of the nation's major religious leaders have written to Senator Frist today to urge him to schedule an immediate vote on the bipartisan stem cell research bill.
The Senate Majority Leader Bill Frist says he's had a political change of heart over stem cell research. Speaking from the floor of the Senate this morning, Frist said he now supports legislation that would expand federal funding of embryonic stem cell research. Frist's reversal brings him into conflict with fellow Republican President George W. Bush. The announcement makes it likelier that Congress will pass a bill easing the strict limits the president set on funding stem cell research.
If we can answer the moral and ethical questions about stem cell research, I believe we will have the opportunity to save many lives and make countless others' lives more fulfilling. That's why we must get our stem cell policy right.
In 2003, President Bush attempted to get a ban on all forms of human SCNT through the United Nations.
A hopeful society has institutions of science and medicine that do not cut ethical corners and that recognize the matchless value of every life. Tonight, I ask you to pass legislation to prohibit the most egregious abuses of medical research, human cloning in all its forms, creating or implanting embryos for experiments, creating human-animal hybrids, and buying, selling, or patenting human embryos. Human life is a gift from our Creator, and that gift should never be discarded, devalued, or put up for sale.
In response, Christopher Reeve presented this testimony.
Now, as representatives to the UN, all of you hold positions of enormous responsibility. You are perceived as the voice not only of the countries you represent, but as the collective moral voice of the world. Millions of patients just like me will be watching with great concern as you continue your important deliberations. Everyone I know of, including the scientists you'll hear from today, opposes the cloning of babies and the pursuit of reproductive cloning. But every leading scientist seeking cures calls for stem cell research to advance. Therapeutic cloning, also known as somatic cell nuclear transfer, offers real hope. The research is still in its infancy, but it has so much promise. And not to encourage the ethical pursuit of this research might result in needless human suffering and destroy the hope of those afflicted with Parkinson's, Alzheimer's, spinal cord injuries, and a host of other conditions for which no cure is yet known. Countries around the world are grappling with this issue and deciding that the purpose of government is to do the greatest good for the greatest number of people. Countries can ban reproductive cloning and still live up to their obligation to provide the best medical technology to all citizens. And I have a real concern that a great medical advance might be lost to humanity should the United Nations recommend a treaty that would prohibit this research. So my prayers are with you, and I hope that you'll make the right decision. A decision based on secular law and morally sound scientific knowledge that will provide hope to millions suffering all over the world. Thank you very much.
The debate over cloning was largely a misunderstanding of our intentions. Not only were we not advocating the cloning of people, we were not even advocating the cloning of individualized embryos. Before 14 days of development, no cells of the body had formed, and the embryo could still split into two or merge into another. Therefore, it was not individualized, and if not an individual, how could it be a person? I saw the twin discoveries of ESLs and SENT as gifts to mankind, tools to relieve suffering on an unprecedented scale. ESLs reminded me of Osler's description of August Weismann's germ-line. This marvelous embryonic substance is eternally young, eternally productive, eternally forming new individuals to grow up and to perish, while it remains in the progeny always youthful, always increasing, always the same. Thousands upon thousands of generations which have arisen in the course of ages were its products, but it lives on in the youngest generations with the power of giving origin to coming millions. The individual organism is transient, but its embryonic substance, which produces the mortal tissues, preserves itself, imperishable, everlasting, and constant. Initial work in the lab was directed toward demonstrating the ability to manufacture to scale only a small subset of the rare and valuable cell types on this tree. In order to begin a human clinical trial, each of these regenerative therapies has to undergo extensive safety and efficacy studies. The first such studies were among the most voluminous submissions in the history of the FDA.
Just 3 days into the new Obama administration, the Geron Corporation got permission from the Food and Drug Administration to start human trials using stem cells.
Cells useful in the repair of spinal cord injuries were moved forward, especially the severe types of injuries that leaves one a quadriplegic, that is, unable to move either arms or legs, or even breathe on one's own. Spinal cord injuries can have a devastating impact on the life of a patient and his or her family. Because the adult human body has little to no innate capacity to repair such serious injuries, some accidents can leave one unable to move anything from the neck down, and in some patients, even no ability to breathe. In a dramatic first use of ESL technology, patients with increasing dosages of cells began to display impressive recovery of function. It is currently too early in the trials to declare success. Nevertheless, it seems unfortunate that Christopher Reeve could not live long enough to see the therapy he championed actually introduced into procedures for patients with injuries similar to his own.
I couldn't drink, I couldn't feed myself, I couldn't text or pretty much do anything. I was basically just existing. I wasn't really living my life, I was existing. And now, after the stem cell surgery, I'm able to live my life. If I was there, and if I was able to thank them, I would just tell them, thank you for giving my life back. Thank you for allowing me to live my life again.
Unless they have blue eyes.
How big is a miniature husky?
Actually, they're about like
In the following years, the proteins within the egg cell that were capable of reawakening telomerase and transporting aged somatic cells back to the beginning of life and back to immortality were analyzed. It became clear that a time machine for aged somatic cells could be constructed without the use of an egg cell. Today, this is commonly called induced pluripotency and is no longer controversial. In 2010, we were able to show that an aged somatic cell could be reprogrammed back to an embryonic state, reversing the developmental clock, but more significantly, that the telomere clock of cell aging could be rewound back or even longer than they were at the very beginning of life. Seeing this result, a practical and facile technique to reverse the aging of human cells, to make it possible to take a cell from even a 100-year-old person back in time, back to the immortality of the germ-line, resetting the clock of cellular aging, such that it was now possible to make young regenerative cells to repair tissues damaged the result of age, was one of the most gratifying moments of my life. Few have fully grasped the impact the science will have on humanity over the coming decades and centuries. For the first time in the history of mankind, we have finally grasped that which is finite, and through human agency, out of love and compassion for our fellow human being, transferred the power of immortal renewal to the mortal soma. We have taken aged cells back to immortality.